Paul. So it's my pleasure to introduce Paul, who's going to give you a lecture now and another one tomorrow on some virology basics, coronavirus, and a little bit of work in R, I think, tomorrow, right, Paul?
Yeah, that's right. Yes.
Yeah, so which probably would have been beneficial like 4 weeks ago for some of you,
Oh,
but we're getting to it when we're getting to it. But Paul, thank you so much for being here and for giving a talk.
you are welcome. I'm happy to be here and hope to do my best to deliver some information to everybody. Can you see am I sharing properly that you can see?
Yep, it looks perfect.
Okay, all right. Okay, hello everybody and welcome. So my name is Paul Cantalupo and I'm a computational biologist currently in the at DBMI with Uma Chandran, my supervisor, data analyst there. I received my BS in microbiology in 1992 from the University of Pittsburgh, and then went back to get a master's degree in information sciences at the University of Pittsburgh in 2001. And that took my career where I was a bench scientist for about the first 15 years where I was doing experiments at the bench, and then launched it into more of a computational throughout the past decade and a half. And now I'm with DBMI doing computational work. So, all right, so let's go ahead and jump right into, so what is a virus? Well, what I'm showing here is not a virus, but it's in a eukaryotic cell that you've probably been familiar with or have seen, where it contains a nucleus with our DNA inside here and organelles and ribosomes that are necessary for translation of proteins. So what else is not a virus? A bacteria is not a virus. It is an organism that's a single cellular organism that has a membrane surrounding it with its DNA inside and possibly pili and flagella that come out from the cell membrane. Now here are some viruses. So they come in some fantastic shapes and sizes. One here, this bacteriophage. Bacteriophages are viruses that infect bacteria so that they will infect just bacterial cells. And they come in and it looks like a spaceship. It's rather complex. And other kinds that are helical, spherical, and polyhedral. And if we take this spherical virus, crack it open, this cartoon diagram of a virus, this shell is a protein coat made up of a different number of protein subunits, and inside is the genomic material. And so that leads to a little bit more of a proper definition of what a virus is, that it's an infectious, obligate, intracellular parasite with genetic material that's surrounded by a protein coat, as I've mentioned, this capsid surrounding, and sometimes a membrane. Let me first get into the word infectious. So infectious comes from infect, and really all that means is to get into. In means into, and fect comes from facere, to put. So to put into. And so really that is just about the ability of a virus to get into a cell. Doesn't say anything about whether or not it could cause disease. And then obligate is that it is restricted to this particular mode of life, that it must be an intracellular parasite. And we'll get more into that later about why that is. And viruses are very small. About 500 million rhinoviruses, which is a particular virus family, would fit on the head of a pin. And I wanted to give some sense of scale for, I lost my mouse. Where'd my mouse go? Wow, I don't know. Okay, I'm sorry. So let's zoom in a little bit. Here's a sesame seed, a grain of salt, down to about 1/4 of a millimeter or 1/2 of a millimeter. And as we go down through, we see an example of a eukaryotic cell, a skin cell around 30 micrometers in length. if we zoom in a little bit more, we start to see our first bacteria here, and a baker's yeast to give you the sense of scale between these two. But no viruses have shown up yet. We need to scroll in to more of a nanometer scale where we start to see HIV, measles, bacteriophage. And where's that rhinovirus that I mentioned? Here it is here, only about the size of your ribosome, where we have literally thousands of these in one of our cells. So the sense of scale is quite vast and amazing to dive into. So, okay, going back, let's also then to mention that viruses are part of our microbiome. And the microbiome is an aggregate of all of these microorganisms like bacteria, fungi, and protists and viruses that are residing on our bodies, on our skin, as well as inside our mouth, inside our organs and tissues. We have probably 10 times the number of bacterial cells in and on our body than we do the number of cells in our body. So the viruses are part of that microbiome. Now, the entire planet lives in a cloud of viruses. So we know that viruses infect every species on the planet that we've studied, that we eat and breathe billions of virus particles every day. Viral genomes are integrated in our own DNA. That one particular family of viruses, the retroviruses, inhabit about 8% of our own DNA. It's pretty amazing. The number of viruses on Earth, just the total population, is massive. Just bacteriophage alone is 10 to the 31st. To put that into perspective, that would be 100 million light years if put head to head. That would reach beyond the Andromeda galaxy, which I think is 2 million light years away. And we are all infected with several viruses right now, you and I, that we may have multiple herpes virus infections, polyomavirus infections, and once infected, it is for life. And usually they don't cause any issues until sometimes when you're immunocompromised or have some other health condition. Now, most viruses that infect us have little or no impact on our health. So you've heard a lot about a lot of bad, dangerous viruses, and of course we'll talk about SARS and that. But there are many viruses really do not impact our health. And why is that? Well, for three primary reasons that most viruses will pass right through us. This red pepper is infected with the pepper mild mottle virus. And when we eat fruit and vegetables or other material, we're ingesting tons of virus. And this virus was shown to be one of the most abundant fecal viruses, that there are billions of them per gram. There are also many good and beneficial viruses, such as human herpes viruses may play a role in suppressing HIV-1 infection. And we also have an amazing immune system that has two major arms. I don't have time to get into those details, but that we have an innate immune response, an adaptive immune response. An adaptive immune response is somewhat represented here by, you may have heard of T cells and B cells. That B cells make antibodies. T cells may just kill other infected cells outright. They're called killer T cells. So we have a pretty amazing immune system that can handle a lot of infections. And I wanted to throw out a poll question to everybody, just to get a sense of what you have taken in from what I've said and get a feeling for this. So which statement of these is true? If you would all try to make a best guess here, that do all viruses make us sick? Can our immune system manage most of the viral infections? Humans are usually infected with just one virus at a time, and viruses only infect a few species on Earth, or viruses are not obligate intracellular parasites.
I believe the poll's launched if you guys want to respond.
Yeah, great. Give it a little bit more time. Yeah, this is all anonymous. I have no idea who's so just answer away. Okay, I think a large majority of the votes are in, and that's great. Most people have picked that our immune system can manage most viral infections, and that's true. That is one of the reasons why most viruses are not going to harm us. Okay, I'll move on then to the next. Let me see here. I think I do that. Okay, so give you some perspective on why maybe it would be interesting to study viruses and why we do study viruses. Since they are obligate intracellular parasites, just the study of viruses would help us to learn about the host that it's infecting, that it's getting into the cells of, so that we will learn about host and viruses. And through that, we have learned very important principles of biology, such as DNA replication, RNA splicing, and many other important processes that go on within our cell have been elucidated, have been determined and figured out because of our study of viruses. And if this SARS-2 pandemic that kind of makes the point for this, our global health really depends upon it to really understand and study viruses. And that we don't really have cures for viral infections, mainly just treatments to prevent and help stop the spread of infection. We can, of course, develop treatments and drugs against disease. One example is bacteriophage therapy. Bacteriophage are viruses that infect bacteria, and here at the University of Pittsburgh has been shown to help fight against very difficult bacterial infections where nothing else was working. So by the study of viruses, we are able to treat that disease. And what is very exciting is that there are a vast amount of viruses that are undiscovered so far. Estimates are that there are about 2 million species on the planet, but we only know about 10,000 viral species. So if you would think of just one virus per species, we still have 1 million viruses to find. So it's pretty exciting. But of the viruses we do have, we have many viruses, 10,000 of them. How do we classify these viruses? So there are two major ways in which we do so. One is through the classical hierarchical system that you may be aware of, that we have our kingdom, phylum, class, order. As we go from the top down there, we're going from more general to more specific. And we, in virology classification, by looking at the nature and sequence of the nucleic acid, of the RNA or DNA in the virion, the symmetry of the protein shell, that the capsid that's surrounding the virus, whether or not it has a lipid membrane or not, we call that an envelope, and the size of the virion. The family of viruses that that we use a convention of viridae at the end. So you may see retroviridae, herpes viridae, coronaviridae. That is the family that virus belongs to. Then the genus is usually just ends in a generic virus ending. So like coronavirus, that could be talking about many different coronaviruses. And then you have a specific species of coronavirus, such as SARS-CoV-2, SARS coronavirus 2. Now, there's another classification to be aware of, is the Baltimore classification devised by David Baltimore, a Nobel Prize-winning virologist about 40 years ago, I think, that from these thousands of different viruses, he was able to group them in a different way into just 7 groups of viruses, as opposed here to these 100 virus families approximately. That his reasoning was that all viral genomes must make RNA that can be read by host ribosomes. Now, why is that it must be read by host ribosomes? Because no virus encodes for ribosomes. So viruses are kind of a distinct entity that they do not have the ability to, number one, make their own ribosomes in order to then make protein. So that's why they are an obligate intracellular parasite. They're parasitarizing, if that's a word. They are a parasite on our host ribosomes so that they can make proteins and then make more copies of themselves. And a little bit more information about this classification. This is a very busy slide. I don't expect you to take all of this in, but I want to just center your focus of attention on this mRNA here. So this is just remaking the point about how all viruses must make RNA. They must get to this point in order for that RNA to be translated by the host ribosome to make viral proteins. And if we back up, we can see many groups or different groups are either composed of a DNA or RNA genome. And I've placed some names of viruses that you might be familiar with. Herpes viruses are these double-stranded DNA viruses. Coronaviruses I've mentioned. Now we see it as a single-stranded RNA virus. And influenza are a negative strand RNA virus. Now, what is this positive versus negative strand? Well, it's by convention that the positive strand of RNA is ribosome ready, meaning that this genome of the coronavirus can be loaded onto a host ribosome immediately for it to be translated into protein. So therefore, there are strategies for these other viruses of how to get to make RNA before they can be translated into proteins. Oh, and please feel free to ask any questions in the chat. I just want to say that. Okay. All right, so another poll question then. So why is mRNA placed at the center of the Baltimore classification? So is it because all viral genomes are made of mRNA? There's really no specific reason. Because mRNA must be made from all viral genomes. Because Baltimore studied mRNA. Or is it to learn important principles of biology? It might be a little bit of a tricky one. The slide before is pretty busy, but think about how viruses need to parasitarize the host cell, and it needs to make some molecule in order for it to be translated into viral proteins through the use of the host ribosome.
And if I can interject, is the poll working for everyone? Can you see results, Paul? Because I see
Yeah,
zero.
I do see. Yeah,
okay. I'm not sure why. I don't see any results, so I just wanted to make sure that everyone had access. Perfect.
Yeah, it looks like we have about 50% who voted, and we had a little higher voting last time. I think this one's a little bit more tricky, but So it is, and most people did get this right, that it is because RNA must be made from all viral genomes, so that each one of these virus groups need to have a strategy in order to get to make RNA, so that then it can be used by the host ribosome, which goes back to because viruses do not make their own ribosomes, so they need to use their host ribosomes. Okay, on to the introduction of coronaviruses then. So with a little bit of some basic virology background. So coronaviruses are members of the family Coronaviridae, and they are spherical in shape, enveloped virions, so that there's a lipid membrane that's surrounding the nucleocapsid, which we will get to. And they are about 120 nanometers in diameter, bigger than a rhinovirus, where many more could fit on the head of a pin, but about 10,000 of these could fit on a head of a pin. It's a linear, single-stranded, positive sense RNA, about 30,000 bases, 30 kilobases in length. And one of the virus proteins called the spike, or the S protein, the spike protein, protrudes from this envelope, and it gives it its crown-like appearance. Crown or corona comes from, crown comes from the Latin word corona, for crown. So that's some of the quick background on coronaviruses. Now,
I have a question.
yes, go ahead.
Do the S proteins, so do they act as the antigen?
Yes, they can. That is correct. That is one of the antigenic peptides or proteins of the coronavirus that our body can recognize to mount an antibody response to. Yes, and we'll get into that a little bit about some vaccine development, about what it's targeting, which kind of is a foreshadowing for that. Okay.
Thank you.
Yeah, you're welcome. So coronaviruses, there are many out there, and they infect a wide range of animals. And I don't just mean, if you might have heard of some of the possible reports of SARS-CoV-2 infecting your dog or cat. No, these are normal coronaviruses that have circulated in dog and cat and bird populations for a long time. So that these are normal viruses that may cause gastroenteritis or hepatitis and pneumonia in various animals. But for us, we have 4 primary common cold varieties with these somewhat cryptic names: 229E, OC43, that just give us the common cold every year, so that we may be infected by one of these viruses this year, by another next year, and then maybe by the same virus in another year, and because we don't mount a lasting immunity toward those viruses. So the common cold and most coronavirus research was, or I should just say that the common cold was just thought to be a nuisance for years. And that the study of coronaviruses was rather boring until 2002, when the first SARS appeared, and now called SARS-CoV-1 to distinguish it from the SARS-CoV-2 that we're dealing with, as well as a MERS outbreak in 2012. And this is the Middle Eastern Respiratory Syndrome virus that infects camels. And SARS stands for Severe Acute Respiratory Syndrome. And these are believed to be zoonotic transmissions, where it's any disease or infection that is naturally transmissible from a vertebrate animal to humans. And another example you might be familiar with is rabies. So rabies, you may come across a rabid animal. Maybe your dog has been bit by a rabid animal, and it has the rabies virus and can be transmitted to you through a bite. And so that's a zoonotic transmission. So a little bit about the history of SARS-1 and MERS, about where they came from. We use the term reservoir of where these viruses are normally circulating and how they transmit to other populations. So that these two viruses emerged as human pathogens through a zoonotic jump from bats here. For SARS, it was through a cat called a civet. That there's many of these in marketplaces in Asia, that outdoor marketplaces where there's a high density of animals and a lot of interaction between humans and these animals. And that the belief would be is that this virus was transmitted from a bat to a civet to a human to have caused SARS disease. And the same thing in camels, that in the about 30 years ago or so, it's thought that a virus from a bat entered into the camel population, and in the camel population that this MERS virus is circulating. And then through the close interaction and contact of people and camels in the Middle East, that there was a transmission. And so MERS is essentially a camel, a kind of a common cold, but that can cause some disease as well, that circulates in camels. And these two outbreaks resulted in about 8,000 cases and 800 deaths in 2002, with a case fatality rate of 10%. So this term case fatality rate is just for the cases that we know about, so that when somebody presents with the disease, or that they're sick, and that it's considered that person, okay, you have SARS-CoV, and so that you are a new case. And that of those cases, there were 800 deaths. And that is the 800 divided by 8,000 for a 10% case fatality rate. MERS had a much higher case fatality rate, so that if you did get this virus, it was much more deadly at 34% case fatality rate. Fortunately, there were not many cases. The virus did not spread that well. It was contained well enough. And these infections were well beyond the that epidemic phase of those viruses.
I have a question.
Yes.
What is it about bats that they're the ones who these viruses originate with?
Yeah, bats are, so one thing to know is bats are mammals like us. Bats have a very large population. I forgot what the number was, but just the total number of bats themselves, it rivals the human population, and that they congregate, millions of them, in caves. And so and so there's a much, I think, better possibility for transmission of viruses among bats themselves. And so that a pool of viruses have really developed within bats, and it even goes beyond coronaviruses, that there's been many studies of bats to show that they're rich in other virus families as well. And so all that combined with our closeness to nature, in a sense, with open animal markets, or with just our interactions with camels in the Middle East, And even here and even in just normal agriculture settings, like working with normal farm animals, that we can get foot and mouth disease from them. And so it's kind of by our, what's the word? By just our culture, or just how we live our lives, that we come into contact more of these viruses.
Thank you.
You're welcome. All right, and so that brings us to SARS-CoV-2, where the virus itself is called SARS-CoV-2. The disease is called COVID-19, for coronavirus infectious disease 2019. Sometimes those terms are used interchangeably. It's similar to or human immunodeficiency virus is the virus name. The disease it causes is AIDS, AIDS. So here, this whole situation developed at the beginning of December in 2019, where a number of patients with pneumonia surfaced, where doctors did not know what the etiology of that disease was. Etiology meaning just what was causing that disease. And that several of the first 4 cases of this virus were linked to a seafood marketplace in Hunan, China. And then the World Health Organization was informed of this pneumonia of unknown cause by the China CDC. And China jumped on this to isolate SARS-CoV-2. And then on January 10th, and this is just amazing how rapidly that we can determine the genome sequence of a virus. People have gotten their PhDs 40 years ago for just sequencing 2 base pairs of a virus. I mean, it's quite amazing. So that within just a few days or weeks that the genome was released. So Chinese scientists were able to get this data and release it to the public. And then everything went a little crazy after that, the pandemic took off. The World Health Organization declared this public health emergency, and we are where we are today now, which is this point here about there being, as of today, looking up on Google, 660 global deaths with 151,000 United States deaths. And just to put that into some perspective, that the flu it usually causes 50,000 deaths a year in the US. And the 1918 flu pandemic killed 700,000 US. So that this virus is rather scary, rather deadly, and I really hope we'll be able to contain it with various measures that we can discuss in a few slides. Okay, and so show you some data to give you some sense of the origin of SARS-CoV-2. You might be wondering, how are all these viruses related? What I'm showing here is a phylogenetic tree of many coronaviruses. So each row, in a sense, each row is a coronavirus, a ferret coronavirus, a mink coronavirus. Here are the human common cold coronaviruses that I mentioned, the NL63-229E. And you can see I'm highlighting next here, the brown arrow, the SARS, the original SARS virus, SARS-1, is very similar to a SARS virus isolated from a civet cat, and that it is related to SARS, a SARS-related virus in bat. So we have not found a virus that is as similar to the civet virus in bats, kind of indicating maybe a jump into civets and then a jump into human. And to give you a sense of what this scale is, how these lines, the distance from, let's say, this point to a virus, it gives you a sense of genetic relatedness to other viruses, is that this distance here says 0.1, which let's say this is about an inch, which is 0.1. It means that there's 0.1 base pair substitutions, or 0.1 substitutions per base. So let's multiply that by 10, which would mean that there's one change in every 10 base pairs in comparing two of these genomes together. So that if you have to travel out so far to here, so that these two viruses maybe would be about 20% different from each other, approximately. And so that's where we can take a look at how the current coronavirus, which this was an old paper, well, yeah, old paper back in March, or I think February of this year, where it was being called the Wuhan virus at that time, but is more correctly referred to as SARS-CoV-2. It is very similar to a virus that was discovered in bat several years ago at about 96% nucleotide identity. And to give you a sense then of how SARS-2 is about 80% identical to SARS-1. So therefore we would be following this line out this way, traveling over here to SARS-1, approximately about 80% identical. And so that leads to the model, just like with the other SARS-1 and MERS, possibly a related SARS coronavirus in bats. Maybe there's an intermediate host, we don't know yet, and that has led to the infection in the human population. Okay, so a little bit about the a blow up of the SARS virion itself. So what we have is a spherical virus. So this has been just kind of cut in half, or if we were looking at an apple, the apple seeds would be in here, then maybe the stem would be sticking out here. That here's the genomic RNA that is it's wrapped in this helical nucleocapsid form. And what I mean by nucleocapsid is it is complexed with the N protein. So this coating of the N protein on the genomic RNA and wound in this helical fashion, this is called the nucleocapsid. And then this entire nucleocapsid is surrounded by a lipid envelope. I think the light blue structure here are lipids surrounding this entire nucleocapsid. And embedded within the envelope are several proteins. One of them is the membrane protein, which provides structural integrity to the virus, as well as another protein called the envelope small membrane protein, the E protein, which makes contacts with the genomic RNA on the inside. And here is the spike protein that is sticking out from the virus. And it has a few different domains of that protein, different regions that perform different functions. And this spike protein is what allows specific interaction with a cellular receptor called ACE2 receptor on human cells. So which allows the S protein to first bind to that receptor and then penetrate into the cell. And so let's take a look of that in more detail. And so this is a broad view of the full life cycle of a coronavirus. So me orient you to the upper left of the diagram, where here's our cartoon of coronavirus with the spike protein sticking out and the ACE2 receptor lining this cell. And the ACE2 receptor is on our airway epithelial cells in our lungs, and this virus can attach to this receptor and then allow for this engulfing and getting inside the cell, where then the viral genome, this single-stranded RNA genome of the virus, is released from this shell. So it's basically kind of cracking open inside, and then this genome is released. And if you might have remembered, this positive sense genomic RNA is ribosome ready. So it can be placed on a ribosome and translated. And one of the first things that is made is the viral polymerase. And that is also called an RNA-dependent RNA polymerase, because it's going to perform RNA replication by binding to the positive sense RNA and then making a copy of itself, the complementary form of that genome, which is a negative sense by convention, that positive sense, and then now we call this the negative sense genomic RNA. And then this process can then continue, copying the negative sense into another positive sense, and doing that many times over to replicate that genome many times. And while that replication is going on, there is also transcription that is occurring from the RNA by the viral polymerase. So it does multiple functions, and it will create what are called subgenomic RNAs. The word is not that important, but the term here is these shorter subgenomic regions. These RNAs are much shorter than the full-length genome, and they will code for those proteins that we talked about. The nucleocapsid protein then goes on to make a complex with the viral genome to form the nucleocapsid. And the other proteins, spike, membrane, and envelope, those were the three proteins that were stuck into the lipid envelope of the virus. They're transported over to the ER, the endoplasmic reticulum, where then this all causes a recruitment to complex with the nucleocapsid into these Golgi vesicles, which create the formation of a mature virion. So what you might notice here is that the envelope, the lipid membrane of the virus, is really taking a piece of a membrane, from our Golgi organelle. So these viral proteins have just kind of been inserted across here, and that the virus will just capture this and encapsulate itself with that, and then that becomes the virus. And then it is released from the cell through a process called exocytosis. And I think we have another poll question here, is back to that spike protein. So why do coronaviruses have a spike protein? Is that to provide structural integrity to the virion? They don't have a spike protein. Spike is to bind to and stabilize the viral nucleic acid, to opt the cellular machinery to make more virus, or is it to bind specifically to a cellular receptor to gain entry into the host cell? So we'll give a a couple minutes for seconds for that. All right, let's one more second. I think we have about a majority of voters so far, so might be a good time to go on. Good, okay. So we do have that most people have answered to bind specifically to a cellular receptor to gain entry into the host. And that, just to reinforce that, that was that spike protein sticking out from the coronavirus that then will bind specifically to this ACE2 receptor to then get into cells. So that raises the point there of if a particular cell does not have this receptor, then it can't infect that cell.
I have a question. So,
Yeah.
like, does the binding of the S protein to the receptor, like, does that like stimulate the synthesis of the viral polymerase, or like, is the epithelial cell like an environment where the viral polymerase can exist as opposed to?
Yeah, I think, yeah, good question. The binding of the S protein to the ACE receptor is not really doing anything to activate any of the replication of the viral genome, but is really just for the mechanism of allowing it to enter into the cell, so that once inside the cell, then that viral genome, in a sense, becomes activated. Then, if you want to think of it that way, that there is this activation of this viral genome to make the viral polymerase to then make more copies of itself, of its viral genome, and then eventually assemble into many new viruses in the end.
Oh, so the fact that like it's like positive RNA can't really get translated by this ribosome into like proteins, is that what sort of triggers the synthesis of viral polymerase?
So it does initially get translated when it comes in to the cell by the host ribosome, and the first thing that's made is the viral polymerase. From that initial, translation event, it creates the viral polymerase, and then that kind of what starts that cascade of making many copies of itself. So maybe that could be a targeting mechanism for some kind of therapy to block the interaction of this viral genome RNA, so that it doesn't bind to the ribosome. So therefore, the virus could infect the cell, but then could not replicate and cause disease in that sense. Okay. All right, so let me put this pretty busy chart here, but I wanted to try to show some overview of where viruses and infectious disease lie on a chart of contagiousness versus deadliness. And along the y-axis is contagiousness, or the R0 number, referred to as the reproduction number. And what that is that if you are sick with a disease or with a virus, it's how many more people are you likely to transmit that infection to. So if you're at 2, you'll give it to 2 other people. Those 2 people will give it to 2 other people. That's 4. So anything above 1 is an exponential increase. We can see here that measles is very, very contagious. And I'll describe a little bit later why that is. But thankfully, it's not a very deadly virus. That would be along this scale here. And this goes back to the case fatality rate that I had mentioned about if there's 100 cases of a particular disease and 10 people die, that would be a case fatality rate of 10%. And that's where the original SARS here is located. And let's look at this, rabies. This is really scary, that if you leave rabies untreated, unfortunately, you do not survive a rabies infection. So it is not very contagious. It does not have a high reproductive number, but it's a very deadly virus. And SARS-CoV and SARS-2 is sort of lying in this space right here. Although research, we've only known about this virus for 6 months or more now. And so more of these types of studies are going on to understand its case fatality rate, its reproduction number. The hope is that it would either go down this way, preferably to the bottom left, certainly not to the upper right, where we become a virus that's extremely deadly and very contagious. Okay. All right, and so a little bit about the mode of transmission of SARS-2 and some control measures. And so this brings up the concept of aerosols versus droplets and fomites. And so let me define fomite, and it really is just a surface. It's a fancy word for how a surface such as a table, your cell phone, that may harbor a microorganism that is just, a bacteria is on the table or a virus is on the table, and you might pick it up and ingest enough of that virus to, if you put your hand in your mouth, to cause infection and possible disease. And droplets, well, actually, let me describe aerosols first. Aerosols are very tiny, small particles that can travel long distances. Here on this diagram, through increasing distance, we can see that only aerosols in these small blue dots are transmitting further away from this infected person than are these larger droplets. And aerosols would be things like cigarette smoke, a perfume smell that you can smell from a long ways away. And so, but droplets, on the other hand, which is the primary mode of transmission for CoV-2, these larger droplets do not travel that far. Hence the reason for the 6 feet of distance, that most of these droplets, as they come out, the trajectory is not very far, and that they fall to the ground. One thing to be aware of, though, is that the amount of droplets and the distance of these things, how far they would travel, really would be, for instance, exhaling, just normal breathing. You're not going to be creating that many aerosols or droplets. But then as you start to talk, you're starting to spew out more droplets. Coughing, of course, sneezing, whenever things are coming out of you much more forcefully. So yeah, just that fact of talking, the droplets are coming out, and saying certain words, like even if you're just trying to tell somebody stay healthy, the way that healthy is pronounced, and it's been shown that many more droplets come out of your mouth just by trying to say something nice to somebody. Okay, and then some control measures, like I was just mentioning there, avoid face-to-face talking. So that is one of the more risky things to do, that if you're really just across the table from somebody and you're face-to-face talking without a mask, just the fact of those droplets kind of flying out of your mouth can be a little bit dangerous. And that's the reason for masks and being physically distant. and maintaining good personal hygiene, you know, if we're picking up things off of a table or a surface, to wash your hands, and for being outdoors in an outdoor space rather than indoors, so that outdoors that these aerosols and droplets have a much broader range to diffuse in the air. And this diagram just very simply just shows that, you know, wearing a mask, we are essentially reducing the amount of aerosol, reducing the amount of droplets, and the distance to which that they will travel from ourselves. That, you know, as we might be infected, then it is a measure at least to reduce these aerosols and droplets. So it kind of just becomes a numbers game. That is, as we're all practicing these measures, we are significantly reducing the chance to spread the virus. And let me just go back. And that's what would help to reduce this contagiousness number. You know, so maybe we can force this number down to 1, to where the virus is not likely to spread then, because we're really eliminating many chances for transmission through these control measures. All right, let me see. Okay, almost done. I think what we may still have a few minutes, I think. So let me give you some sense of testing for current and past infection. And to start out by saying that the general course of infection would be along the x-axis here in days after start of symptoms. So that, let's say you start developing symptoms on day 0. But even before that day is when you're initially infected. So maybe 5 days ago, a week ago. And then this blue line is representing the amount of virus as days go on. So that is, you're increasing in viral amount, another term, viral load, viral titer, that before you even know that you're infected, you have generated a very high titer of virus in your body, which is one of the reasons why this virus can spread a lot, or spread easily, is because unfortunately you don't know you're sick, and you're passing the virus to other people. For something like the flu, it's usually you're most infectious when you really feel like you're sick. You're like, oh my gosh, I'm sick. I know I got to go lay down. I need to stay away from everybody. I'll come back out of my room after 2 days. But with this virus, you're highly infectious before that point. And so then after those start of symptoms, virus replication peaks and then starts to wane. As the immune system starts working, less virus will be detected by this initial nucleic acid test, which is usually given whenever you feel like you're sick, you have some symptoms, and that you take this nucleic acid test, where a cotton swab is put up your nose to get a sample of your upper airway epithelia, and then through an experiment is called PCR to test for the nucleic acid of the virus to see if virus is present. One thing to note about this is we are really only detecting nucleic acid in this test. We're detecting the viral RNA. It does not necessarily mean that we are not actually detecting live virus. But it definitely correlates with a higher amount of this nucleic acid, that it's a live virus infection. There's just been some issues with low amount of detection of the virus in these PCR tests, and doctors wondering, well, okay, we have a very low amount of nucleic acid we're detecting. Is this person infectious or not? You know, we don't know, because they could just be detecting remnants of the genomic RNA, but not any real live virus. And then as the immune system starts to work, antibodies in the blood will ramp up after about 10 days. And then an antibody test can be done later on after infection to see if we have mounted an immune response to the virus, if our body has been exposed to the virus. Interestingly, though, here, we don't know how long these antibodies will last, nor how protective they are. That's important because there's reports of COVID parties where people get together to try to get infected by this virus, where somebody might think, well, hey, once I'm infected, I'm never going to get infected again with this. We absolutely do not know if that's true or not. We don't even know if the second infection could be worse than the first. So just because you may have antibodies doesn't necessarily tell you about reinfection. It just kind of gives you a sense if you have already been exposed to the virus. And then I think finally, just to end up with just some overview of vaccine approaches for the virus. There's a link in the lower right-hand corner, that COVID-19 vaccine tracker. There's over about 200 different vaccines that are in different phases of development. Two of them here, or actually three, companies Moderna and Pfizer are developing a nucleic acid vaccine, and Oxford is, this group is developing a different type. With the side on the left here, these nucleic acid vaccines, it's really just composed of a piece of RNA that you're pretty familiar with now, with just this is not exactly the coronavirus genome, but it's a piece of RNA that does contain a piece of the coronavirus genome that encodes for the spike protein. And this is then encased in a lipid coat, which is kind of almost like a mini virus here that these companies are making, but these are called lipid nanoparticles that are stable enough to be then injected into your muscle, where they can fuse with cells, get into a cell, and since it only contains just the one protein from the virus, it's not going to cause any infection, it's not going to cause any danger to the person receiving the vaccine, and it will express that particular viral protein, the spike, and an immune response can be mounted to it. So these two vaccines are currently, I think, in phase 2, starting into phase 3. It's a really long track to get a vaccine approved and finished. But they are on an accelerated path to hopefully have something by next year. A lot of things could happen in the meantime, though. And then on the other side, I think for the sake of time, I'm just going to say that there are just other ways you can make vaccines. And this is kind of a different way, which I think is good. We just need all these different vaccine approaches to try to be developed to compete with one another to see which virus or which vaccine would be most effective. And so I think that is what I wanted to finish with. And I just want to conclude and say that viruses are everywhere. They're out there. There are new viruses that are emerging every day by natural processes that we need to be aware of and know about, so that is through our understanding of viruses, this will hopefully help us prevent future pandemics. And we need smart, motivated people like all of you to become virologists. So think of that as a possible career path for yourselves and to help save lives. And will you be the next one to defeat the next novel coronavirus? Or maybe it might be another coronavirus, or any other novel virus that's going to cause us problems. So I just want to then end by thanking my supervisor, my boss, Uma Chandran here at DBMI. I'm part of her lab, the UC lab. We are a genomics analysis core. And for the members or for David and Solomon and the Hillman Academy, to thank them for this opportunity. My previous boss, who I worked for almost 30 years, was Dr. Jim Pippis here at the University of Pittsburgh. My friend Eric Palenko for helping me with the lecture. And Vincent Racaniello, who is a professor at Columbia. He has some wonderful podcasts called This Week in Virology. So if you're interested in following up with more virus information, he has a great podcast every week, multiple episodes a week. They've really been talking about SARS a lot, of course, for the past 6 months. And his virology lectures, which are free to all online, very detailed, good lectures that he has out there. So I just want to thank you for your attention and your interest in this topic. Any other questions?
Thanks, Paul. That was excellent. Any questions, guys? I know you're probably tired after now 2 hours on a Zoom call.
Yeah, thank you for participating in the polls, everybody, and asking the questions throughout. Thank you. All
right, if there are no more questions, well, you'll have a chance to talk with Paul again tomorrow if you have any questions. So
we'll try to do some analysis of COVID data in R.